In contrast to presynaptic mechanisms, postsynaptic modulation of synaptic transmission and strength can be achieved by a remarkable variety of neurotransmitters and receptors. Before we focus on the details, let's briefly remind ourselves of what we learned already about postsynaptic excitation and inhibition. Whether a neurotransmitter causes excitatory or inhibitory effects at the postsynaptic neuron isn't a property of the neurotransmitter molecule per se. The neurotransmitter itself is a signal to act, but what action the neuron takes depends on what postsynaptic receptor molecules receive that signal. The postsynaptic receptor determines what effect a given neurotransmitter has, and the nervous system is characterized by a relatively small number of neurotransmitters, but an extreme diversity of different kinds of receptor molecules that interact with those neurotransmitters. Generally, we can differentiate between two major types of postsynaptic receptor molecules, each with very distinct mechanisms of action and very different speeds of operation. These two broad classes are ionotropic and metabotropic receptors. First, let's consider ionotropic receptors. They can induce fast changes in the postsynaptic membrane potential on a timescale of milliseconds, or thousandths of a second. Structurally, these ionotropic receptors are generally ligand-gated ion channels with a binding site specific for a specific neurotransmitter molecule. The neurotransmitter is like a key that opens or closes the channel, allowing ions to flow through. Upon binding the neurotransmitter, ionotropic channels open a central pore that allows ion flux across the cell membrane, and this ion flow modulates the membrane potential directly. In contrast, metabotropic receptors operate at much slower speeds. These receptors are not ion channels at all, in fact. Metabotropic receptors induce changes in the electrical and biochemical properties of the postsynaptic neuron in the range of hundreds of milliseconds to seconds. Since they're not ion channels, they don't allow charges to pass through the membrane directly. Instead, they act indirectly through the activation of intracellular signaling cascades in the postsynaptic cell through the activation of so-called second messenger molecules. These second messenger signaling cascades are sequences of molecular interactions. One molecule causes a change in another, which interacts with yet another molecule, and so on and so forth. The inner workings of cells are made up of intricate networks of interactions between molecular signaling cascades, or pathways, and metabotropic receptors interface between the synapse and this aspect of the intricate inner life of the cell. Signaling pathways can instruct the cell to alter its basic functions, including what proteins it produces. The relationship between a neurotransmitter and its ionotropic or metabotropic receptors needn't be exclusive within a single cell. The same neurotransmitter release can have multiple effects on the postsynaptic cell, each acting on a different timescale. Many of the classic fast-acting ionotropic neurotransmitters, as we'll see shortly, can also act as neuromodulators through metabotropic receptors, both presynaptically and postsynaptically. As such, a single neurotransmitter release can act almost immediately, while also having slow, long-lasting actions on the timescale of seconds to minutes, sometimes even weeks. These intricate, nested interactions over many timescales add additional layers of complexity to neuronal signaling. This complexity plays a critical role in enabling the nervous system to change and adapt over different timescales.